Okay, well, I think we'll make a start. It gives me great pleasure to introduce Bruce Robinson to you today. Professor Bruce Robinson's from University of Western Australia. He's a little bit of a polymath. I've just reading his bio this morning. He speaks 5 languages. I don't know how fluently he speaks each one of them. Has 3 children and 2 grandchildren, and he runs the Fathering Project at UWA, and he's written 6 books on fathering. So that's the important stuff. Now, the less important stuff, he is the author of more than 200 papers, numerous book chapters, review papers for prestigious journals, and I'm just going to read a few things here. He's the director, I won't have done that one.
Yeah, sure.
Okay, sorry. It's long. He's scientific director of NHMRC Centre of Excellence. He's won various medals, including the Wagner Medal, Premier Science Award, the TSA&Z Medal, the AMA, RSCP, and Saints Award. In 2013, he was made a member of the Order of Australia and was named both West Australian of the Year and WA Australian of the Year in 2014. And I understand, more importantly, that he's an ex-football player and also football coach for UWA. And I'll hand it straight over to Bruce.
Yeah, thanks very much, and I do apologise for being late. There's great advantages being a physician-scientist, but there are great disadvantages. One of them is you actually have to carry your phone when you're on call. So I left without and had to turn around and go back again, so I do apologise. This is actually more science than physician today, so this is about tumour neoantigens, a bit of a hot topic, and we're collaborating with some people here on this, so it's kind of fun. You can see lots of friendly faces in the crowd, fantastic. So this is some current stuff we're doing. But you know, you can interrupt me anytime if you like so. So this is me, so I'm a lung specialist, with another CAT scan there. So that's the origin of thinking about some of these things. So we're talking about cancer tumour immunology. And when I prepared this lecture, so now given a few times this year, in some form or another, I actually pulled out an old CT scan. So this is this computerized sort of slice, if you like, through the lung. But this is from 1989, our hospital. And just to orient you, this is the spine here, spinal cord. Where you see black, it's lung, and there's a big tumour there. This patient got a recombinant alpha-2A interferon in a clinical trial we did. And after 3 months, the tumour shrank. And the patient lived for about 5 years, but their life expectancy was 5 months. And I guess it was kind of fun to pull it out because this is one of our early clinical trials that made us realise that immunotherapy could work under some circumstances. But being a little bit OCD, and as a trained immunologist, I asked the question, what are the antigens in there that are causing the immune system to do that? The reason we assumed it was the immune system, of course, was that the patient had lived for 5 years. And this happened to quite a few patients on that trial. So this is really about what are the antigens. What does the immune system see? So we're talking about Neo-Antigens and cancer immunotherapy, and mostly we'll just talk about new data. Okay, so I'm in the tumour immunology group at UWA, and we do a couple of things. Tumour antigen cross-presentation is something we've done a lot of which we've done a lot. I'm not going to talk about that. And chemoimmunotherapy. I just mentioned that because they're the 2 things that people think about when they think about our work. I'm not actually going to talk about them. But here's the thing about immunotherapy. So as I said, I'm a chest physician. And we've been beating this drum for a long time. And there's 2 things that I think we have slightly got right in the crystal ball gazing. One of them was that immunotherapy would work for lung tumours, including lung cancer. So we work a lot with 2 carcinogen-induced tumours, mesothelioma caused by asbestos and lung cancer caused by cigarettes. But no one believed that immunotherapy would ever work. And kept telling us it was rubbish. Anyway, so this is what you would see. So I used to go and present at the World Lung Conference, the World Lung Cancer Conferences, and when you had a session on cancer immunotherapy, this is the sort of audience you get. And they'd usually be the presenters, you know, some junior postdoc and then worker maybe, whatever. and then they'll all get up and present, and then they'll leave. And it's pretty humiliating to chair these. But anyway, 18 months ago, I chaired the World Lung Cancer Conference session on lung cancer immunotherapy, and from the podium I decided to take a photo. I couldn't even fit everybody in. So what's different? Two things are different. The first is that tumour immunotherapy is now working, and this is so-called checkpoint blockade, the brakes on the immune system which antibodies can block, particularly these two. And might say particularly PD1, but probably both of them. And the journal Science called it the breakthrough of the year before last. But the other thing is, which I want to talk about today, is the ability to identify tumour-specific antigens by next-generation sequencing. So this is already in the clinic, this one isn't, and this is sort of what's kind of fun to talk about. So just going back to immunotherapy, this is non-small cell lung cancer. So just let me say that when you have advanced non-small cell lung cancer, your life expectancy, depending on what stage it is, but it's measured in months. And this is AntiPDL1 therapy, not from us, it's from New England Journal. And this is called a spider plot, so it's what happens to the lesion. So if it goes down like that, it's shrunk and stays shrunk, and if it goes up, it's just not responded. You don't need to be a statistician to know that this stuff is working pretty well, and this is like a year here, and then number of patients still doing very well. So it's just very interesting. It's very interesting and exciting times in cancer immunology. But we're actually, I mean, if everybody did this, we wouldn't need to find any neo-antigens. We wouldn't even be doing these experiments because, you see this? You wouldn't need to be doing these experiments because if everyone did that, it would be great. But they don't. And of course, they have temporary responses to it. So we're interested in how to turn the failures, which is actually most, into successes. So just to remind you, there are 3 carcinogens that are known. So most human cancers, there's no known carcinogen, breast cancer, stomach, etc. No defined carcinogen. But there are 3 ones that we do know about. One is sunshine and skin cancer, melanoma, squamous cell. Cigarette smoking and lung cancer, and asbestos and mesothelioma, but that also causes lung cancer. So we study asbestos cancers a lot. For one reason is that they are important for health. I mean, it's not a very nice cancer to have. It's going to cost $300 billion in compensation. There's a lot of worried. Well, Australia has the highest incidence in the world, and Asia has an impending tsunami because they import over 1 million tonnes of asbestos every year. But also it's quite a good model to study cancer for various reasons. So this is, as you've already seen, a mesothelioma. So the asbestos comes in through the lung, penetrates the lung, irritates the pleura, that's the casing of the lung, and after between 15 and 50 years, you get a tumour there that grows around. Crushes the lung and invades the local organs, can invade out through the skin as well, and they get the cancer syndrome, they lose weight, get tired, etc. Median survival 9 to 12 months, so it's not a very nice cancer to get. We have the world's highest incidence in Western Australia. Oh, here you go. This is when I was a medical student, it was a rare disease, and then this sort of thing happened. This happens everywhere where asbestos has been used, which is most of the Western world. A couple of kids in our famous picture. A couple of kids playing what looks like in the dirt, but in asbestos. The stuff on their face is asbestos. And there's quite a few incidental exposures, which about everyone's been exposed to asbestos. You talk to anyone, they'd say, oh, you've been exposed to asbestos. I mean, everyone in this room's got asbestos in their lung. It just comes from living in an industrialised society. But these kids obviously had a higher dose because you know, just playing. Both of them died in their 30s of mesothelioma. So we have a national centre of excellence, as Alex said. So why is it a good model? Well, it's one of the few animal models where if you put in asbestos, it's a pretty good mimic for the human cancer. In fact, it's almost identical. And you can't say that sunshine doesn't do that, and you might say smoke it does. It's very hard to get a good animal model of human cancer, and this is one of the rare ones. But also patients, because you know who's been exposed, you can study them very early and track them. And we've published a biomarker which is okay for that. Also, there's other good things. For example, there's fluid. It produces fluid with tumour cells. And that's the fluid outside the lung. So this is the chest X-ray. That's the white thing's the heart. This is fluid, malignant effusion, and there's tumour cells in there, which provides a number of advantages. One is it's accessible, so you can get the tumour out. It's representative. I can come back to that if you want to. And you can sample it sequentially, so you can measure evolution of resistance and things like that. So there's all sorts of reasons why it's a good thing to study. Anyway, let's get on to the topic of the day. So when I talked about the first CAT scan, I talked about the fact that I was interested in knowing what the immune system was seeing, in other words, what the antigens are. And target tumour, there's only really two sorts of target tumour antigens. One are the self-antigens, and the other sort of the Neo-Antigens. Self-antigens, of course, which have been there all the time, but, you know, hidden or whatever, but nevertheless, relatively hidden. So MAGE and tyrosinase and all these things are studied. And we looked for them at one stage, and we didn't find much of them, but we made an intellectual decision that it was extremely unlikely that the human thymus would be fooled into thinking that something that was expressed later on ought to potentially be immunogenic. Just a priori, we decided that even though we didn't know what the antigens were, that we would focus on the Neo-Antigens. Neo-Antigens are the ones that are just happening in the cancer. They have never been seen by the host, the thymus, before. Mutations, mostly. But the question is, how do you study them? Back when we made this decision, which, as I alluded to before, we feel quite proud about, how do you study them? Well, one is, we didn't know what they were back then, so it's about 20 years ago, so you have to model them. And we put in various model Neo-Antigens into tumour cell lines, viral HA, albumin, and other things. And then you can leapfrog them. I'll talk about that maybe, and then identify them, which is the topic of the day. So for modelling, we have these asbestos-induced tumours, which, as I said, are good models for the human tumour, pretty good models anyway. And BALB, black, CBA, put all sorts of things in them. We've got a transgenic mouse that we've made, and we study other tumours as well. So these mouse models are essential to work out how the host engages with the tumour, such as what are the initial events, how does it evolve, how does it escape, and when you give therapy, where it might be failing. This is like a party political statement, because I'm a clinician, as I said, and I go to meetings and they say, well, you don't need to study mouse models anymore because this treatment's working in people. I said, it's working in some people, but when it fails, which it mostly does, how are you going to know? How are you going to know what's going on? How are you going to know the rules of engagement? Anyway, there's no doubt that you need to continue to study mouse models. Actually, the way we started this was, again, looking at those CT scans of the tumours, asking ourselves, what do we know about how the host immune system engages with a tumour? Before any manipulation, like the patient walks in, there's a scan, there's a tumour, how does the host immune system engage with that? And the reason we made the decision about neoantigens is because we already knew there were lots of mutations. So a mutation should, if you like, a mutation that is coding, should make the tumour look at least a bit like a virus to the immune system. The immune system, of course, doesn't know it's a virus, it's just receptors, but it should make it look a bit like a virus. So how the host is engaging with a tumour, you need a mouse model for that. Now, I do need, this is where I just switched to the teacher in me, but I have to do this because people who aren't used to talking about tumour immunology don't understand maybe or don't grasp the different steps. So this is the 7 steps required just for a CD8 T cell to destroy a tumour cell. And the reason for talking about it is that if you don't understand where something is blocked, like I said, most patients don't respond to this new therapy. If they do respond, then fine. But if they don't, at what point have they failed? Point number one is the tumour has to have some neoantigens in it. And then point two is they have to go to the lymph node and be cross-presented by the dendritic cells, and in all of our studies, almost entirely restricted to the drained lymph node. Doesn't matter about the assay, really. But that's not enough either, because you have to have a T-cell repertoire that responds appropriately, expands, and circulates. And now you've got circulating anti-tumour T-cells. This is what we measure. But you've only got to step 3 in these 7 steps. Step 4 is, I mean, what's the point really if all these tumour-specific CD8s are just dripping around in the blood? I mean, they don't know where the tumour is. So, especially because you can take out the primary, but you've got to seek and destroy the little metastases. So how the heck do they know where they are? So they've got to traffic to the tumour. Once in there, the evidence is, and this is the weakest, what's probably fairly strong evidence now, that the CD8 T cell will not just enter the tumour and attack it. It probably needs to, when antigens, limiting, etc., be re-engaged by a host APC, re-stimulated. This is could be a problem. And then it can go on and attack the tumour, which has to express, of course, the class 1 beta microglobulin and the antigen, and that's one way it can escape, by blocking expression of the target, as happens in some viruses. But then in this system, which is not a virus, it needs, PD1 blocks this step, it needs help. So CD4 cells can either help, arrow going up, or hinder Tregs. Anyway, these are the 7 steps, and when something fails, you've got to understand where it's failed. That's the point of animal models, and I think like people give some treatment and they measure number 3 and they say, oh, isn't that great, we've got some CD8 T cells, and isn't that great, we must fix the problem, but of course there's 4 more steps after that. So we study all of these things. Just an aside, so we modelled them. You can also leapfrog them. The idea of leapfrogging them is this. Here's the tumour again. And then this time after chemotherapy, it shrank. And this is the notion of letting the immune system find the antigens. Why do you need to know what they are? And I kid you not, I looked at this CT scan one day, and I looked at it and I pondered and I thought, that's a lot of tumour to die. Where have all the antigens gone? That's what I thought about. And in those days, the dogma, transient dogma, I've got to admit, if dogma is transient, can it be still dogma? Anyway, the prevailing notion was that apoptotic cells tolerise the host repertoire. So I had this brilliant idea that the reason immunotherapy was failing was that we were giving chemotherapy, delivering a lot of apoptotic cells into the cross-presentation pathway and tolerising the host repertoire. That's why you couldn't get a response after that. Like a lot of my brilliant ideas turned out to be complete rubbish. Because, in fact, it delivers a lot of antigen. I won't go into the details of this, but basically it delivers a lot of antigen into the cross-presentation pathway in the lymph node and in the tumour. And this is what generated the idea of the tumour as its own vaccine. You may never need to know what the antigens are. That's a point we could discuss later on if you like. So the idea is that an apoptotic cell would load the DCs, the CD8s would expand, and then they would load the cross-presenting APC in the tumour, which is a bit defective, at least in our models. Then it provides an opportunity for CD4 help in the distal site. Anyway, we've translated that, but I want to get on to identification of these things. So the only reason they can be identified now, so neoantigens, lots of mutations, we model them, we leapfrog them, but hey, you can at least try to find them now. So I know a lot of you will know a lot about this, but there's different ways you, this is because you can sequence the mutations and identify them. So a couple of machines here, Illumina and IonTorrent, both of which we've used. And in humans, when you sequence cancer, this is now a famous figure from Nature, I think it was. Anyway, it's what we call the mutatogram, and it's the average number of mutations per megabase. So on the right-hand side, you can see it's about 10 on average. And this is melanoma and lung cancer, the two of the three carcinogen-induced tumours. And then you go down, and then you get right in the middle, you get a lot of common tumours. Ovary, melanoma, prostate, breast down here, pancreas. A log less on average. And then you get down further, and you get these different other sorts of leukemias, some pediatric cancers. And it's kind of interesting because the ones up here tend to respond best to this checkpoint blockade. In other words, there's already some drivers there, so if you take the brakes off, it can work. The challenge is how to make all these guys respond. So we've sequenced mesotheliomas, they're kind of in the middle, so the third carcinogen doesn't induce quite so much in the way of as the point mutations. So I'm just going to have a little physician-scientist moment. This is whole genome sequence, circus plot, major structural variations and things. It doesn't just how you represent whole genome data. But here's the point. We got this from our collaborators, and we had a teleconference. It's one of the first four we did. And I picked it up and I looked at it, and I kept staring at it because she was my patient. Right? She was 45, and I remember she sat in my office. I remember exactly where she was sitting with her daughter, and cried and all that sort of stuff. Give the news. She lived for 2 years, did fairly okay. 2 years, and then she died. And I was looking at her circus plot, and it's like a spiritual moment for a physician scientist. This is like a Rosetta Stone. This is the Rosetta Stone of why she died. Right? So it's kind of for a physician scientist who can look at these things and think it through, and then see the patient. It's kind of cool, to be honest. Anyway, let's go on to the mouse. So I mentioned we have lots of different lines. So the way we get these lines is give the mouse asbestos intraperitoneally, generate cell lines. We've got lots of them. And as I mentioned, for some of them we've put in artificial Neo-Antigens. In this case, HA. This is AB1HA. Just to make a point, we don't put them in to make them immunogenic. So this is not it is immunogenic, but it's not protectogenic, if you like. In other words, when the animal rejects this tumour, it's protected against this one, and vice versa. This is what you would call a spy antigen, or was one of the words we use, a spy antigen. It just kind of reports back on what's going on without actually changing what's going on. And just to, again, make the case that asbestos-induced mouse models are similar to the human, the average single nucleotide variants as mutations in 15 mesothelioma mouse lines in 3 different strains is 450, whereas the other mouse lines that are commonly used in these studies are more like 1,500 to 3,000. So these are off the scale when it comes to that mutatogram. And this is probably 30 rather than 10. So this presented us with our first question, I guess, which is, I already told you that the most responsive tumours are the ones with the most mutations. So mesothelioma is kind of around the 1 mark. So this is obviously a lot less than the ones in which, the few papers in which this sort of thing has been described already. So is this too low? That's the question. Would there be no neo-antigenic reactivity? So we took the tumour and sequenced it. So let me just ask the question, who here would be familiar with the DNA sequencing? So we've got a few here. So this is going to sound really obvious to you, but just so we don't lose people, I just need to explain it. So this is just exome sequencing, i.e. the bit that codes for proteins, and we're looking for antigens, so you're only interested in the first instance in the exome, which enables you to sequence deeper. We also did RNA-seq, so that's sequencing the RNA, which helps you to look not just for expression, but you can cross-verify your exome reads and also look for splice variants and fusions and what have you. So it comes out in this kind of FastQ format, and then the binary alignment map. So for those who are interested, we aligned it to a MM9 as our mouse reference genome, which is actually a black line, black mouse line. So you have the normal against that reference genome, and then you have the mutated tumor. The third step is to call the variants, because you can't just can't read it like a book. So we've used 5 different variant detection programs. 3 of them are statistics based, and 2 of them are heuristic. Heuristics like the best guess, practical way of handling lots of information. So we all use heuristics every day to make a best guess for when we see people. I mean, you've got 2 arms, 2 legs, I assume you're a human, etc. That's a heuristic. Something's with a trunk and leaves, you think it's a tree because it's a best guess and it's a practical way of doing things. So I'll show you what happens when you get sequenced. And wouldn't it be great if everything gave you exactly the same information? But this is what happens. So here's our tumour. And when you apply these different variant callers to them, you get this kind of thing. So this one here, for example, SAM-TILS, which is a heuristic from Human Genome Project. There's 607 calls outside that no one else has called. 209 here. This is a Canadian one. This is from the Broad, which is a famous institute, and it was probably the world leading institute in this field in Boston. 111 out there. I mean, you'd be a bold man who called 111 from the Broad wrong. And then there's 250 in here, so then there's 2 calling it, so you think, how are we going to deal with this? Okay, why am I mentioning this? Because the whole point of this is to translate it into patients. So right now, we, that is the team, have made a lot of progress, and this isn't really being done in people yet. I've just come back from 3 weeks overseas discussing it with colleagues. What's the best way to do it in people? Well, one team has done it, I talked to them, but done a bit. But if you're going to vaccinate people to try to stimulate the host immune response to a neo-antigen, which antigens are you going to put in your vaccine? Because if you get it wrong, well, let's say you say, well, we're going to have 3 of these 5 callers. How do you know that there's 250 plus 111 at the Broads? Actually, this has improved since then. But anyway, that these 361 haven't got the gold in them. Be bold leaving them out. We left them out. So we just, to start off, pick the ones that are in the middle. Called by all of them. I'm not sure that's a good idea, to be honest, but anyway, you've got to start somewhere, and you can't test everything. So we pick these ones. And then, of course, you want to work out what genes they are, etc. And then, of course, when you're going to go to that much trouble, you need a validation step. And so you're saying a sequence, which is the gold standard. Because of our selection process, basically everything got validated. So, but anyway, so this is the pathway. So we did RNA-seq, and then alignment, which I mentioned, which is these are the alignment processes for RNA-seq from Hopkins. And then expression, I mentioned, another part of this tuxedo suite from Hopkins, variant callers, and then selection of candidates. So the way we did that was we got our tumour, of course, compared to normal genomics, so we can work out what's a mutation and what isn't. And then we also looked at the one that had HA in it, that is this cell line that had the viral antigen stably transfected. And total exonic SNVs, non-synonymous. And then we looked at the ones that were common, because as I said, one cross-protects against the other. So again, we're just trying to focus in on the most relevant antigens. We then picked those that had a thing called a Fred score, which is the probability of the variance being correct. So Fred 60 means there's a 1 in a million chance that you got it wrong. We set the bar a bit high here. I mean, 30 would be 1 in 1,000 chance. So. And then we picked a few more that were present in 3 of the 4 data sets. That is in the exome. Anyway, we picked 57. And then of course, you've got to have an affinity cutoff for your mutated peptide, which I'll mention. The mutated gene has to be expressed. So if it's only in the exome and not in RNA-seq, you're wasting your time because it's not an expressed mutation in that cell line. There's not going to be any protein there. We actually picked 10. FPKM is fragments, but it doesn't matter. It's a measure of how many reads do we, how much mRNA do. It's a bit like a Northern blot, really, or a microarray. Anyway, the point is it has to be expressed. We picked 10, possibly could have picked 1. Other people do that. Anyway, ended up with 20. 20 to start with. So let me just reorient it to tell you where we are now. We've identified DNA mutations. We've identified those that code for a mutated protein, and it's in RNA-seq as well. But we haven't yet shown that anything binds to that anything is loaded into the class 1. In other words, the mutated peptide being presented, nor that any CD8 T cells can actually recognize this mutated peptide. All we've got to is here. And this is a crucial thing when you think about patients. So, you know, if you think about it, I mean, let's assume for a moment that some sort of vaccine using mutated peptides will enter the clinic in some way. I think that's almost certain. How are you going to pick them? I've already told you problems, you know, that the 361 that we left out from the Broad's variant caller, right? So how do you know that's not where the money is, right? How are you going to pick them? And here's the other point. Should you use the CD8 T cell to pick them? In other words, to see what the T cells are already seeing, but weakly. Just to increase your chances of finding something that might be useful in people. So the way you do this, and again, I know I can show you, this is like old hat for you guys, and a lot of other people here too that would be familiar with peptide prediction. So just again, just so that I know, who here is not familiar with how you get mutations and look at peptide prediction, etc. Well, who is familiar? There you go. Maybe that's the best way. That's a few people here. So I'm going to bore you guys again. So anyway, the way to work it out, here's the mutation, and because class 1 can bind anything up to between 8 and 11, you have to go 10 on either side to create all possible sequences around the mutation to then load into a peptide binding software. So this is NetMHC Pan 2.8. We've actually got the same results with IEDB, but there's half a dozen of them that are used, or more actually. But anyway, you load them all and you load 5,000 at a time. And you find out, you get a grid. So this is, these are the 3 restriction elements, class 1. 8 MERS, 9 MERS, 10 MERS, 11 MERS, wild type, mutant. Wild type sequence, mutated sequence. So when we started this, we thought that the best antigens were going to be those that changed an anchor residue. In other words, that something that didn't previously bind to the class 1 molecule now did bind, because that's kind of where a lot of people have told us the money would be, a lot of people elsewhere. But what Shokan Marty, who's a student, he picked all the anything that bound, and asked the host. I've already told you how he selected them on expression and FPKs and affinity. But anyway, he asked the question, fine, you pick your top 20, but you ask the question, is there any evidence that host CD8 T cells are engaging with any of these? So to do this, he took pills of 5 peptides and did a gamma interferon ELISpot down here. It's more red down here. And only 20 hours. So I just mentioned the 20 hours because remember that, so the animal has the tumour, 2 weeks later, the spleen or lymph nodes are removed, and it's only a 20-hour culture. That is, you're not stimulating with any of these peptides for a week. You're only asking, is there already a T cell reactivity existing in that host against those peptides? So you're setting the bar very high. We've already set the bar high on our selection. Now we're setting the bar high on the T cells. So a lot of people would culture them for 5 days. We don't do that. So we didn't do that. So we're already really heavily biased against finding anything at all. 20 hours. So this is the result of that. So on this panel, so these are the 4 pools, each with 5 other peptides. 5 of, so that makes up the 20 peptides that we originally screened. This is the wild type peptide, and then this is the mutant sequence. So these are peptides now put into an ELISpot. Again, you don't need to be a statistician to work out that pool A's got nothing. Pool B looks as though it's got something in the mutant pool. Pool C is a self-reactivity because the wild type also reacts. In fact, that wasn't a consistent finding. Pool D has nothing as well. So you have to look at that and think one of those 5 peptides in pool B is a mutant peptide that the host is seeing reasonably strongly that they can do it in 20 hours. So 10 out of 10 in Group B. Group C, I said there was a bit of self-reactivity only in a minority of experiments. So we deconvoluted pool B, the 5 peptides that made up that pool. And again, you can see the first 2 didn't do much and the second 2 didn't do much, but this one here, UQCRC2, is where most of that reactivity, well, probably all of it actually, is to be found. So this is a mutant antigen in this tumour. It's in both the tumour and the HA transfectant. It's expressed, and the host T cells not only can see it, but they can see it after just, it's a pretty strong antigen actually. What is it? It's a component of the respiratory chain. It's actually involved in aerobic metabolism. And it's not an anchor residue, it's proline to alanine at the 4th position in. And pretty highly expressed, and it generates a hydrophobic side chain, which actually might be pretty important. Now, it's a mutation in the aerobic metabolism pathway. When we discussed this in the lab, myself and one other guy, Ian, think this is great, and everyone else thinks, you know, they roll their eyes and yawn. But Ian and I are interested in a thing called the Warburg effect, where cancer cells seem to prefer anaerobic metabolism even when there's plenty of oxygen. And it's some sort of survival advantage. So this looked to me like it could be a mutation in something that gave the cancer a survival advantage. And the attraction of that is you can't escape from it, because then you can't escape by immune selection. Anyway, I haven't managed to stimulate a lot of interest from anyone else other than Ian, but anyway, we keep thinking it's a great idea. Well, of course we can. We will try and immune select against that and see what happens. But here's the problem. Here is an obvious problem. I've just told you that it's high affinity, or certainly highly expressed. I've forgotten what its affinity exactly was. Anyway, it's at the top of the table, near the top of the table on affinity and highly expressed, and it's like a pretty good antigen. And you saw the ELISpot, 20 hours, and you think, great. But here's the problem. The animal has a cancer that's growing. I mean, if you do this in people, you find antigens this way, you say, excuse me, but you are finding failed antigens. You're finding failed antigens. Is it just a weak antigen? In other words, are we finding what is basically just a weak antigen? So we did a simple thing. We actually just compared it to the viral antigen. So these are 4 mice, early spots again, no peptide. These are the controls, wild type. At that time, we were looking at NB4. This is the mutated peptide. All 4 mice responding. I mean, some more than others, as is usual in mice. You've already seen this. But when we looked at the viral strong antigen, it was the same. In other words, it looked like it wasn't a weak response. Well, at least it wasn't a weak antigen. It might be a weak, it's not a strong response, so it's not strong enough to get rid of the tumour. But it is a strong antigen. It's as good as viral HA. Good as a strong, dominant viral epitope. So it can't be that this is just a weak antigen that's escaped selection and, you know, sort of fooling you. So here's the problem. We had 20 good reactivities. And I told you how we chose them. We picked the best 20. I was sure we could find more than one. But we only got one. 1 out of 20. Does that mean there is only one? Is it like a virus that's going to keep up with this huge dominance? But viral dominance is probably different. Oh, we don't know. But again, this is the problem for patients. How are we going to translate this? How are we going to choose it? 20 good predictions, 1 reactivity only. Are the other 19 complete lemons? So one possibility was that there would be competition between strong antigens. Because you pick the strongest or we pick the best candidates, maybe there's competition. So we didn't do it. Well, we already know that there's no competition between our antigen and HA, right, in the same tumour. So there's 2 strong antigens, no competition, no obvious competition. They both lit up in that previous experiment. But it turns out we'd already done an experiment to test this. And this is Christine Bundell, 9 years ago, did this experiment. She put in a polytope transfected. This is with Delia Nelson. So took Lewis lung and put 4 dominant epitopes in, 3 viral and the dominant albumin epitope, and an in vivo CTI, just to see if they would all show up or whether only one would show up. And this is an in vivo CTL. So when there's a peak that is reduced, that is in vivo killing. And I'll put some asterisks there to help you out here, but these are the different viruses, etc. But the point is, 3 of the 4 showed good killing. We don't know why this one didn't, but 3 of the 4 showed good killing. So 3 out of 4 is pretty good, right? So we're only getting 1, remember. So it doesn't, you wouldn't predict, therefore, that, I mean, it could still be competition, but why are we only getting 1? So again, is that it? Is there anything we can do to bring out the other 19? So there's 2 things I'll mention. One is Treg depletion, one's immunogenic chemotherapy. So Scott Fisher has got these mice in. These are BALB, FOXP3, DTR transgenic mice. So basically, the mice are completely normal, but the Tregs, that's the FOXP3, the cells that regulate, suppress immune responses and express FOXP3, have the diphtheria toxin receptor. So if you give them diphtheria toxin, you just kill the Tregs. That's the point. So you clean out the Tregs. And the question is, if you took out such a powerful suppressive cell, would that unmask some of the other 19? I figured it's going to unmask half of them, maybe 6, 8, 9, or 10. So just to prove, so this is the protocol. 2 shots of DGX doesn't, the mice are totally happy. Treg cells disappear, other CD8, CD4s, nothing happens to them. Here's the thing, in a dose-dependent way, the tumour also disappears. Take out the Tregs, 10 nanogram per gram. This is a Kaplan-Meier survival plot, so this is a good response, no response. Right? So there's a dose-dependent effect. more Tregs you take out, tumour actually goes away. So, and also, the other thing that happens is the black bars, Tregs, deplete them, they bounce back within 2 or 3 days, and of course stay up. But the CD8s, which are supposed to be controlled by the Tregs, ICOS positive activated CD8s, they are let off the leash and stay high for another week. It's interesting when you think about how this might be working. So, the UQ2, which is the one we've already shown you, has an increased response, but everything's increased. What we've shown you about the ICOS positive CD8s. But another antigen, GANAB, has appeared out of the 20. But none of the other 18. So GANAB is an alpha-glucosidase, again mutated at position 4, this time a positive to an uncharged side chain, again fairly reasonably highly expressed. So we found 1. Now out of 20 we found 2. We're not sure about the GANAB now. It may just, we're not sure how much of it is Treg depletion, how much of it is time, or a combination of both. Anyway, it's a more complicated discussion than it needs to be. And I've already told you about immunogenic chemotherapy. And so we asked, does immunogenic chemotherapy, which as I said kills the tumour cells and delivers a massive load of antigen into the lymph node, surely that'll unmask a few more? It does not. It increases the response to the one we already know about. That's mutant, as I said, used to be called NV4. But no response to any of the other 19 peptides. So this is that leapfrog strategy again, trying to make the immune system respond. So how might this be translated? So here's that patient with an effusion again, and you'll remove the tumour cells. How to expect to translate it? We're in the process of pulling this together, but we haven't submitted any ethics, etc. It's quite expensive. We've got to get it right. Well, the first thing you do, well, the patient will go home, go to the clinic or whatever, if they have an operation to get the tumour, they'll recover from the operation. You'll do exome and RNA sequencing, and obviously MHC typing. Give you predictions of mutated peptides, and then generate a vaccine. The vaccine will consist of some of these mutated peptides, long peptides. Whether you have to use 2 or 10 or 20, we don't know. And the other question is, which other therapies does it need to be used with? Is checkpoint blockade, which is the hot topic, is that the right therapy to combine it with? There's an argument that it's not. Indeed, Bob Schreiber last year in Nature, late last year, published a paper showing that if you use the vaccine of Neo-Antigens, 2 Neo-Antigens, very similar to our data, a strong one and a weak one, or an easy to find one and not so easy to find one, he didn't even need checkpoint blockade, the tumour shrank without needing checkpoint blockade. Anyway, so this is the pipeline. This is why we're doing all this, because this is what's going to have to happen in patients. And then the patient will come back to the clinic and probably 2 or 3 months down the track, and there'll be vials of peptide with adjuvant, and they'll be administered to the patient in some form, which we will have to work out with lots of animal studies. What's the optimal way to give it? But and I'll finish with this, which neoantigens are we going to utilise? I've already been talking about that the whole way through. So this is how we talk about it. There's the ones that you can see, that is, the ones that are seen by the T cells already. And UQ2, the first one, is an example. Use the T cells to tell you which ones to use in your vaccine. But the second one, the second antigen, wasn't seen. We had to manipulate the animal to see it. So that's what we call submarine antigens. They're hidden, but they can be brought to the surface. That's GANAB, the second antigen I showed you. But there's another argument that says, well, why do you need to know? Why not throw the whole flotilla at them? And you might even want to leave some of these out because they might have established some form of negative regulation already. This is all yet to be worked out. It's very new stuff. So why do I think that there's more antigens to be found? I mean, maybe, like in this animal, right, one strong one and another weakish sort of one out of the top 20, but we've actually extended to about 40 now and still haven't found anything in the next 20. I should have said that. And we're going to push through to look at the whole, I think, 80 candidates, but I don't think we're going to find much. This is just single nucleotide variants, point mutations, right? There's lots more things that could be there. So why do I think there are more to be found? Well, I'll just show you this little bit of clinical data from Mel McCoy. So she did a study with Anna Noack and Richard Lake, and they're looking at CD8 T-cell responses after chemo. So here's a patient who comes in, and these are QEII-67 proliferating CD8 T-cells here. So this is normal, right? And then they have chemotherapy and they get smashed down, as you would expect. And then a week later, 2 weeks later, maybe 2 weeks later, they're proliferating again. They rebound, you know. And you think, oh, this is just homeostatic proliferation, who cares? But she did a very interesting thing. First of all, she noticed that some of them were class 2 positive, CD38 positive, and BCL2 low, which actually suggests antigenic stimulation. Doesn't prove it. You look for all the memory antigens and couldn't find any response. But here's the telling piece of data. She looked at these numbers and she split them into those that had most and those that had least. That is, those that had above the median QEII-67, the best proliferative response and the least good. And then looked at their survival. The ones that had the most T cells rebounding had greater survival than the ones that did not. And it's nothing to do with, you know, being sick or anything like that. It's actually to do with something to do with the immune system. As it rebounds, it presumably sees antigens. And currently we want to, let's go back to the previous one, we want to get in there and see what their host reactivity to Neo-Antigens are as compared to theirs. So there's probably potentially anyway a universe of potential tumour Neo-Antigens, and we've just found a couple. And to be honest, we're a little bit lucky to find them so quickly. There's lots of people who are looking for these things and not finding them. So maybe we'll still need to use leapfrog strategies. So to finish, from all of our animal work, what could we predict would be the case in patients? Well, firstly, natural human carcinogens can induce a strong neoantigen response. So the only studies in mice have been with methylcalanthrin, which is a very powerful carcinogen. That's why they've got 3,000 mutations. This is asbestos, which is this is the first study of a natural human carcinogen, which is why we were so worried that it might not generate anything at all. It does. Didn't talk about that, didn't talk about that, didn't talk about that actually. Okay. So I'm going to honour a few people here in particular. So let's go backwards. So Shokang and Michelle are both students, PhD students, and sorry, Sophie. Sophie does all the sequencing. Shokang did all the RNA-seq analysis, pretty much what I've just shown you. And Michelle does the early spots. Those who didn't talk about his work actually did some of the cross-presentation work, which I left out. I say this because Sophie's 1 year into a PhD, and she's got one paper published already, and 4 sitting on the, we had a supervised meeting, and there are 4 papers sitting on the table to put in about sequencing. I mean, she's a very smart person. And Shokang actually did most of this work in his honours. Very bright student, so we're very lucky at the moment. And just to honour too the other key people here, I haven't talked about all their work, but this is the leadership team at the top. Didn't talk about Jost's work either. And I'll just mention a couple of people here. Look, Mark Watson and Shay Leary have been fantastic collaborators and helped us to think about that part of things, and we do part of the work down here as well. And lots of collaborators, but the main thing is that we're interested in translating this into patients. Anyway, thanks for listening. Please fire any questions at me.